So today's speaker is Professor Guy Denuault from the University of Southampton. So I had the pleasure of meeting Guy for the first time there pretty much a year ago, and had a really good time. And we actually spent some time talking about this topic specifically. So I'm very keen to hear his presentation. So thanks for coming on and giving the webinar, Guy, and over to you now. And so I'll switch off my camera and we'll be talking to just blank screens for a while, and then I'll switch on my camera at the end, and we'll do some question and answers. So over to you.
Thank you very much, Michal. It was very kind of you to invite me to your series of webinars on electrochemistry, and I'm really grateful. So what I want to present is some of the work that we've been doing using sample current voltammetry. It's an old technique, but I think it has offered some new opportunities, hence my title, An Old Technique Offers New Tricks. So before I forget, I want to acknowledge the work, starting with Leila, who worked with me quite a few years ago, and then Sam took over and developed a lot of the sample current voltammetry, and then later on Oliver contributed quite a lot, and then I had Sibel, who was on placement from her university in Montreal, she came to my group, and I would be presenting some of the results from all these people. So I want to acknowledge some of the funding as well as colleagues from Montreal who have been helpful, and we're collaborating to develop the applications of sample current voltammetry. So without further ado, I want to get into the topic of sample current voltammetry, but first a little bit of nomenclature. I will be using the concept of SCV, and I want it to mean sample current voltammetry or sample current voltammogram. In the literature, you will see SCV also stands for staircase voltammetry, but this is not what I will be talking about. So a little bit of outline for the talk. First, I will give you an overview of the concept of sample current voltammetry, and then I will spend most of my talk about applications where I used sample current voltammetry with micro-electrodes, and then sample current voltammetry with electrodes which are unsuitable for micro RD experiments. And time permitting, I will talk a little bit about sample current voltammetry for systems, redox systems, where we have adsorbed species but no mass transport. So the concept of sample current voltammetry is actually quite simple. You identify a potential window of interest and you perform a series of potential steps. So these are your potential waveforms, series of target potentials within the window of interest. And for each target potential, you acquire the current transient. So you'll have a family of current transients. And
sorry, Guy, to interrupt. Your slides are a little bit cropped. I'm not sure what's happening there. The left-hand side, looking at it.
The left-hand side. Okay.
Yeah.
Do you mean? Is that better?
No, it's the exact same. Maybe just go out of the full screen view for a moment.
Do you want me to unshare?
Yeah, maybe unshare and start again soon. Yeah. Unusual glitch. Yeah.
All right, strange. Where can I? Okay, do you want me to share again?
Yeah, we'll just see what happens then. Yeah.
Did that make a difference?
Just 2 seconds now, it's coming up. No, it's the same. Maybe you could go out of the full screen mode and see if we can see the full slide then.
Oh, it's visible now correctly.
Is it okay for you, Wojciech?
Yeah, it's working.
Yeah, we can see it perfect now.
Okay, so it's my computer that's the problem. Okay, that's fine. Okay.
Okay.
Yeah.
So just to start again with the concept of sampled current voltammetry, it's fairly simple. You identify your potential window of interest and you perform a series of potentials to different target potentials within that window of interest. And you acquire a family of current time transients. And the idea now is to sample the current time transients at particular times to create what we call a sample current voltammogram. So this is a very old technique. You can look it up in Bard&Faulkner. It's actually a very useful technique to understand the relationship between current potential and time. But what's interesting is that when you do this, you reconstruct these sample current voltammograms, and they have all this sigmoidal shape, but of course the magnitude of the current varies with the sampling time. So very, very simple in principle. When you plot the current as a function of time, you get the series of chronoamperograms highlighted in red here. And when you repeat this for the values of your target potentials, then you can see that your chronoamperograms are changing shape, especially around the wave that was recorded on the voltammogram. But what's interesting is sampling at a particular time. There you recreate these sample current voltammograms, and this is really what we're going to be discussing and how we have applied that to investigate redox processes. So, oops. I just wanted to flag up an old technique which is still available on some electrochemical workstation, which is normal pulse voltammetry, which essentially shares the same potential waveform. So sample current voltammetry and normal pulse voltammetry simply involve going back to the rest potential, stepping to a target potential, and then going back to the rest potential, and so on. And then you increment through to explore the potential window of interest. The big difference is that the signal acquisition in sample current voltammetry is over the whole duration of your potential step, whereas in normal pulse voltammetry, it's just sampled once. So normal pulse voltammetry will give you one sample current voltammogram, and this is available with most electrochemical workstation. With sample current voltammetry, you've got the ability to sample your current transient anytime during the duration of the step. And so you will end up with a family of sample current voltammograms. So now I want to move to applications of the technique, how we have investigated redox processes at micro-disc electrodes using sample current voltammetry. So now the question is, given that you end up with a steady-state diffusion layer at a microelectrode, how are the current potential time data set going to look like when we move to micro-disc electrodes? So first, I just want to have a brief overview of the current time relationship at microelectrodes and how the voltammograms derive from that. So if we look at the diffusion regimes at a micro-disc electrode, at really, really short times, you've got an almost planar diffusion layer, very, very thin. And on transient, now in the planar diffusion where the current is inversely related to the square root of time. And if you were to do a voltammogram in that time scale, then you would get the traditional shape for the voltammogram corresponding to semi-infinite linear diffusion. If we now perform experiments somewhat later, so now we are in the quasi-hemispherical diffusion regime, you can see that the diffusion layer thickness has changed shape. The edge effects have altered the shape of the diffusion layer, and the current transient is now curving. And if you were to do a voltammogram on that sort of time scale, then you would see the shape of the voltammogram tending towards a sigmoidal wave, but with some peaks on the forward and the reverse scan. And if we do the experiment later, now we've got a very well established hemispherical diffusion field. The thickness of the diffusion layer is about 7 to 10 times the radius of the microdisc. Now the current transient approaches a steady state, and at very long time reaches the steady state. And if you perform voltammetry on that sort of time scale, you've got a linear sweep, sigmoidal shape with very little hysteresis between forward and back. So the voltammetry at the conventional voltammetry at microdisc electrode is complicated because we get essentially 3 shapes of voltammograms depending on the diffusion regime. And now what I want to show you is what happened when we perform sample current voltammetry at microdisc electrodes. So these are real sample current voltammogram. It's for the reduction of ruthenium hexamine. So this is the current, this is the potential, and we have here sample current voltammograms corresponding to sampling at 500 millisecond, 200 millisecond, 100, etc., all the way down to 2.5 milliseconds. So what we see is that the sample current voltammograms have a sigmoidal shape, but obviously the magnitude of the current is increasing as we decrease the sampling time. This was the shortest sampling time that we could use with the electrochemical workstation. It's a PGStat 101, so a fairly conventional electrochemical system, nothing fancy there. What we do next is to normalize the sample current voltammograms by dividing the whole experimental data set with the theoretical diffusion control current that we can calculate with the Marwin-Oldham equation. I will show you in a second. So by normalizing the experimental currents with the theoretical currents using n equals 1, what we end up with is normalized sample current voltammograms. And what you observe is that on the whole, they all collapse onto the same sigmoidal wave, and they approach normalized current of 1, except for a bit of noise corresponding to the shortest sampling time. So the SCVs collapse onto a single sigmoidal wave, and that's quite interesting. How did we do the normalization? Well, we divided by a theoretical expression. There are 2 in the literature. You can either use the Shoup and Szabo equation, which is a one expression for all times, which is not so accurate from the millisecond to the second region, or you can use Marwin-Oldham equation, which is a 2-term expression, but it's more accurate in the time scale of interest. So we're normalizing our experimental sample current voltammogram with this theoretical expression. I don't know if you noticed, but to perform these experiments, we've been using a conditioning waveform, which basically involves scanning the potential a few times. These were all done on platinum electrodes, so we went a little bit positive to form some oxide, and then we went negative to reduce to remove the oxides and adsorb hydrogen. And then after a while, we came back and we rested here to avoid the oxide, to hydrogen, and to rest long enough to allow the concentration of the species to recover their bulk value. And at that point, we perform the potential step to acquire the chronoprogram. The reason we do this is because one of the interesting points of the sample current voltammetry is that we can perform some conditioning before every potential step, so that every point on the sample current voltammogram has the same electrode history, and that will be important for some of the reactions that we're looking at. So this is evidence that the role of the conditioning waveform has a role to play. So these experiments, these are sample current voltammograms which were performed with the ruthenium hexamine again, but this time without conditioning. And so what we observe is that without conditioning, there's a systematic increase of the normalized current. And essentially what we see is that the shorter the time scale and the more extra current we get. So this is related to the reduction of platinum oxides which are left on, and if you don't perform the conditioning waveform, you're not able to retrieve the expected diffusion control response, so you're getting extra current. The other important aspect is the acquisition conditions. So these are sample current voltammograms taken at 2.5 milliseconds with 3 different conditions. So the PG-start 101 with a high stability mode gives us this limiting value, which is obviously too small. The PG-start 101 at high speed mode gives us a value which is much more reasonable. And then if we go to a more fancy equipment, the PG-start 30 with a fast acquisition card where we could acquire every 10 microseconds with a high speed mode, then we get the limiting current expected for that particular time. So the normalized current now is equal to 1. So clearly the acquisition conditions are important. These are transient experiments, so you want to make sure that your instrument is capable of measuring currents at short time in a reliable way. So you have to make sure that the response time of your amplifier doesn't affect the currents of the sample current voltammograms. Now, I want to show you how we've exploited the sample current voltammograms to reveal kinetic information. So we've taken a slowish redox couple, and we've performed the sample current voltammograms and sampled the current as before. And what we've done is to normalize the sample current voltammograms and compare the shape of the normalized sample current voltammograms. And what's interesting is that whereas they all reach the same normalized current plateau, the rising part of the wave is now clearly moving from these potentials at long sampling times to these potentials at short sampling times. So what we see here is that the normalized sample current voltammograms reveal the kinetic limitations arising from the rate constant for electron transfer for this particular couple. What we did next was to go back to the literature and take another expression from Keith Oldham's papers. This is an expression that he had developed to analyze the shape of linear sweep voltammograms, disc electrodes, but in the steady state. And he was using this shape to extract the kinetic information, electron transfer kinetics from the steady state voltammogram. But what we did was to replace the steady state mass transfer coefficient that was in Keith Oldham's equation, and we replaced it with a time-dependent mass transfer coefficient, which we could derive from the equation I showed you before. And so we rewrote Keith Oldham's equation, but this time using the time-dependent mass transfer coefficient, and that gave us an equation for the current as a function of potential and as a function of sampling time. So with this, we had the ability to calculate the theoretical sample current voltammogram to compare with the experimental sample current voltammograms. And we did this, and we went further than that. We did a global fit of all sample current voltammograms simultaneously with the theoretical equation. So the symbols correspond to the experiment, and the lines correspond to the theory fitted, where we fitted all the sample current voltammograms at the same time. So these correspond to different sampling times, so long sampling times and then short sampling times here. And from this global fit of all the potential time current data set values, we extracted the electron transfer kinetic parameters from the experimental values. So this modified equation turned out to be quite powerful. We then went on to validate the experiments, the fits with consoles. So what we did was to take the values of the kinetic parameters derived from the fit, and we then used them to simulate sample current voltammograms for the microdisc electrodes, and we overlay here the simulated sample current voltammograms with the thick line and the experimental sample current voltammograms with the symbols. So that to us was an indication that this modified expression was actually quite useful and could be used to model sample current voltammograms for systems with slow electron transfer, for quasi to irreversible electron transfer cases. So now what I want to do is move on to another case. This time we're not going to look at simple redox system. We're looking at the oxygen reduction reaction. And again, we're going to use sample current voltammetry to look at the OR at microelectrodes. So when you perform chronoamperometry at microelectrodes in the diffusion control region, you get a current. So these are current transients recorded at a microdisc electrode in presence of oxygen. And what you find is that at long times, the experimental current is in agreement with the theoretical current. But at very, very short times, the experimental currents are showing a lot more current than predicted under the basis of pure diffusion control. So when we replot this data as current over in terms of 1 over the square root of time, we can emphasize the short time region. And you can see that the experimental current, which is here, is a lot larger than the predicted current. So I'm going to call that the extra current. And if you look at in this region, we could call this the extra charge if you want. If you perform the experiment in absence of oxygen, so with argon, for example, this is the background current that we're getting here. So essentially, below 100 milliseconds, the experimental oxygen reduction current gives a lot of extra current, which is not accounted for by mass transport. So we looked at the oxygen reduction reaction and performed sample current voltammograms over the whole potential for the oxygen reduction reaction. And the sample current voltammograms plotted here demonstrate that the extra current affects the whole of the potential window. The whole of the oxygen reduction wave is affected by this extra current. But what we observe, and this is plotted in terms of normalized current again, what we observed is that at long sampling times, the sample current voltammograms collapse onto the same value, and then at short sampling times, we see a lot of extra current. So since we're normalizing by setting n equals 1, that means 1 electron, this scale is akin to the n apparent. So getting an apparent number of electrons of between 2 and 4 on a microdisc electrode is totally normal. You never get 2 or 4, it's always in between, and this corresponds to the rapid diffusion of hydrogen peroxide. So the apparent number of electrons is never 2 or 4, it's somewhere in between, and it depends on the radius of the microdisc electrode. So a very, very small microdisc electrode would have, in the steady state, would have an apparent number of electrons around 2, whereas a big microelectrode would have an apparent number of electrons of 4. So this was a 25 micron diameter electrode, and this apparent number of electrons is consistent with what we find in the steady state. But what we observe is that the apparent number of electrons for very, very short sampling times is totally unrealistic. And this, again, is evidence that the extra current is affecting the response in this region. This is done with the conditioning waveform. So every potential step in this potential window has been performed with the same conditioning waveform. So every point on the sample current voltammogram corresponds to the same electrode history. So we've analyzed this, and I won't go into all the details, but I just wanted to show you when we perform these experiments on microdisc electrodes, but instead of using a normal platinum microdisc electrode, we perform the experiment with a carbon microdisc electrode, which we had coated with platinum nanoparticles. And when we look at the sample count voltammogram, so again, this is normalized, so this is like the apparent number of electrons. What we find is that even for mild sampling times, the extra current is massive. It's a lot bigger than we would expect. And the analysis or interpretation is that we have several contributions. We've got current coming from the diffusion of oxygen towards the electrode, but we also have extra current coming from the probably remnants of oxide, which haven't been removed completely by the conditioning waveform, but also contribution coming from the electrochemistry of carbon functionalities, probably. And the reason we think the carbon functionalities are involved is because when Oliver performed these experiments, this time with carbon, which had been coated with platinum nanoparticles, so, of the order of 800 nanometers. Now the sample current voltammogram looked a lot more reasonable. The time we could go to much shorter time scale. So this is now a sample that 100 milliseconds, and you can see that the apparent number of electrons is converging towards 2, which is consistent with the fact that we now have a very high mass transfer coefficient on this little electrode, and so the hydrogen peroxide diffuses very quickly. So the apparent number of electrons is close to 2. When we shorten the sampling time, again, we see extra currents corresponding to unrealistic apparent number of electrons. And this is an indication that, again, we are probably reducing some oxygen species that haven't been completely removed during the conditioning. But the big difference with the previous sample current voltammograms is that we are able to go to much shorter times without the contribution of the carbon functionalities, because the whole of the carbon electrode is protected with glass except for the very end, and this has now been coated with platinum. So we have probably lost the contribution from the electrochemistry of the carbon functionalities. So it just goes to show that when you perform these experiments at very short times, then any extra contribution will be picked up, and so you must be aware of that when you perform your sample current voltammetry. So to try and draw a conclusion for the use of the microelectrodes, what we've demonstrated with sample current voltammetry is that whereas the conventional cyclic voltammograms have a very different shape depending on the time scale, sigmoidal at very short times, and then conventional shaped voltammogram sigmoidal at very long times, and then conventional shaped voltammograms at very short times. It's very difficult to compare the shape of these voltammograms as you change the sweep rate here. When you do sample current voltammetry, the voltammograms all look sigmoidal, and by normalizing them, you can reveal some kinetic effects which were not observable when you had if you perform a conventional cyclic voltammetry. So doing the sample current voltammograms actually removes the time dependence from the mass transport, and it enables the comparison of voltammetries which have been irrespective of the diffusion regime. So the sample count voltammogram is quite good in that respect, in that it removes the distortion coming from different diffusion regimes, and it allows us to observe kinetic limitations, or in the case of the oxygen reduction reaction, it allows us to observe how the apparent number of electrons is changing with time scale. So a little bit of advice. You need to be careful that there is no convection. Christian Amator has published articles showing that even on microdisc electrode, you can be affected by convection. So make sure that your longest sampling time isn't too long, so that convection doesn't distort your sample count voltammogram. You must make sure also that there isn't any bandwidth limitation, so make sure your shortest sampling time isn't too short. And of course, you must make sure that the instrumentation is capable of operating at short times. And in particular, what you need to do is to select a current sensitivity which is not too sensitive, because you really want to avoid saturating the current amplifier at very, very short times, because the current amplifier takes quite a long time to recover after saturation. So this current sensitivity shouldn't be too high. So now I want to move to the second topic in the applications, and it's the use of sample current voltammetry in situations where the electrode material does not allow us to make a rotating disc electrode or a micro-electrode. So the idea is to use sample current voltammetry as an alternative to either the rotating disc electrode or a micro-electrode, and perform experiments which will produce pseudo steady-state voltammograms. So this is the sort of cell that Sibel was using to perform sample current voltammetry on thin films, platinum 4s and platinum single crystals. And what I want to show you is how we've been able to use sample current voltammetry to investigate the electrocatalysis of these surfaces. First, what we wanted to demonstrate is the parallel between sample current voltammetry and rotating disc electrodes. So the objective of the next slide is few slides is to show you how we can select sampling times to achieve the same limiting current as you would obtain with a rotating disc electrode. So the idea is to work out a methodology to determine the sampling time that will give us the same mass transfer coefficient as you would obtain with the rotating disc electrodes. So we're going to find the time such that the mass transfer coefficient on the sample current voltammetry is equal to the mass transfer coefficient on the rotating disc electrode conditions. So this is the expression of the limiting current expressed in terms of the mass coefficient, and this is the mass transfer coefficient for the rotating disc electrodes, and here is the mass transfer coefficient for the sample current voltammogram. So this is a time-dependent mass transfer coefficient, which corresponds to here the particular sampling time. So if you select a sampling time, you have a fixed mass transfer coefficient. If you equate these two, you can work out the sampling time for the corresponding rotation rate omega, and what you find is that the sampling time is basically an inverse relationship to the rotation rate. So for ferricyanide in 1 molar KCl, the correspondence goes like this. For a 100 RPM rotation rate, you need to select a sampling time of about just under 1 second. And then as you increase the rotation rate, then you obviously have to select shorter and shorter sampling times. But these are not very difficult to obtain with even very simple electrochemical workstations. So with minimal equipment, you are already able to achieve the sampling times that correspond to fairly fast rotation rates. So you can use sample count voltammetry to achieve the same mass transfer coefficient as you would with a rotating disc electrode, but just bear in mind that at some point you need to, you can't operate at shorter sampling times, because otherwise you will start having distortions from your from the charge and discharge of the double layer. So we would normally make sure that the sampling time is at least 5 times larger than the time constant for the charge and discharge of the double layer. 5 is more than 99.9% completion of the charge and discharge of the double layer. So this is the sort of value that we would select. So these are comparisons between rotating disc electrode experiments and sample count voltammograms. So this is performed for the ferro and ferricyanide in KCl. So we have the traditional rotating disc electrodes for a range of rotation rates, or slow rotation rate, and then increasing the rotation rate. And then if you plot the limiting current against the square root of the rotation rate, you would get the traditional relationship there, linear relationship. So having selected sampling times to achieve the same mass transfer coefficient as those rotation rates, Sibel performed these sample count voltammograms, and the dotted line here indicates the limiting current that you would obtain on the basis of the rotating disc electrode, whereas the symbols show you the sample count voltammograms obtained. And you can see that there is a very good correlation, provided that these sampling times have been selected, as I showed you before. And if you take the limiting current, but this time plotted as 1 over the square root of time, again, you observe a nice linear relationship from, as expected, for the mass transport conditions. If we look at, for example, the voltammogram at 900 RPM, that's the lines, and the sample count voltammogram selected at 94 milliseconds, you see there's an excellent agreement between the two. And again, if we look, for example, at 1600 RPM, corresponding to 54 milliseconds, we again find there is an excellent agreement between the sample count voltammograms and the rotating disc electrode voltammogram. So this demonstrates that if you select the sampling time properly, you can obtain sigmoidal waves on the sample count voltammograms, which are in agreement with those you would obtain with a rotating disc electrode. So it is possible. So I should say that when the SCVs were recorded, the electrode was not rotated, so it was under complete quiescent conditions, obviously to avoid any convection here. So this is just to show that in the sample count voltammetry, we achieve the same diffusion layer thickness as in under hydrodynamic conditions, but we do this by selecting the sampling time to achieve the same mass transfer coefficient. So now what I want to show you is how we've moved from having demonstrated that we could use sample count voltammetry to perform experiments as if we had a rotating disc electrode. We moved to a system which is obviously much more complicated, the oxygen reduction reaction. So this was a platinum disc, so polycrystalline platinum disc. On the left-hand side, we've got the experiments performed on the rotation. So rotating disc thermogram for oxygen reduction at low rotation rates and then high rotation rates, and then plotting the limiting current against the square root of the rotation rate, we get the linear relationship. When we perform the same experiments on the quiescent conditions, but with sample count voltammetry, using sampling time selected to match the mass transfer coefficient obtained with the rotating disc electrodes, we find again a sigmoidal wave with increasing current as we sample to shorter and shorter times. But we observe that there's a bit of extra current in the foot of the wave, and when you go to really short sampling times, what you find is a peak here and then set 2 extra peaks there. So Sibel performed the experiments again, but this time in absence of oxygen, in nitrogen. She used these sample current voltammograms as background and subtracted them. And when you subtract the sample current voltammograms obtained in nitrogen, you find now that these background subtracted sample current voltammograms are much more in line with the currents obtained with the rotating disk electrode, which corresponds to these horizontal dotted lines. But even at short times, we find there's a little bit of a remnant of the features that we saw before background subtraction. So again, very good agreement after background subtraction, but the contribution has not been completely removed. So there's still evidence of surface processes at these short sampling times. So we wanted to see how far we could push the sample current voltammetry. And oh yeah, let me show you this one first. When you compare the sample current voltammograms, which are the symbols, with the rotating disk electrode voltammograms, what you find is that the sample current voltammogram agrees with the cathodic scan of the normal voltammogram. And this is indicative of the fact that there are remnants of adsorbed oxygen species, despite the fact we've used the conditioning waveform to try and ensure there was as little oxide as possible on the electrode surface. There's probably still sufficient amount of oxide left, and you can see that the sample current voltammograms are more in line with the cathodic sweep than with the anodic sweep. Sibel tried to play with different rest potentials to see if the rest potential had any impact on the sample current voltammograms, but this is performed in acidic conditions, and in acidic condition the oxygen reduction wave overlaps with oxide formation reduction, so it's actually pretty much impossible to start from a rest potential which is devoid of adsorbed oxygen species. So we wanted to see if we could push the analysis of the sample current voltammograms further. And so what Sibel did was to plot the inverse of the current against the square root of time to create an analogous to a Kutey-Kilevich plot, as you would do with a rotating disk electrode. So at long times, we see that the data falls onto a straight line, which corresponds to diffusion control. And then there's deviation from that straight line, which corresponds to the extra current due to the presence of the reduction of the oxygen species. But if you extrapolate the linear relationship obtained at long time and look at these intercepts for the different potentials that were selected, and you plot these intercepts on a Tafel plot. So this is now the potential, and these are the intercepts. You find that the sample current voltammograms or the chronoamperograms recorded at the different potentials provide very good agreement with the data obtained with a rotating disk electrode. So in gray, we've got the rotating disk electrode, and in black, we've got the original sample current voltammograms, and then in red, we've got the data coming from the background subtracted sample current voltammograms. So there's a difference in Tafel slopes, because in sample current voltammetry, the electrode ends up spending a lot more time in the oxide region than during a sweep. But still, the agreement is quite reasonable. So we wanted to push this further and see what else we could do with sample current voltammetry for the oxygen reduction reaction. So Sibel carried the switch to platinum foil. So this is where she used this cell. So it's a polycrystalline platinum foil, which is there, contacted from the top, and the solution comes from the top, and it's pressed against a support. The sample current voltammograms were performed with this conditioning waveform, and Sibel investigated two conditions. In black, sample current voltammograms with half molar sulfuric acid, and in red, 50 molar sulfuric acid and 0.1 molar potassium perchlorate. The idea was to see whether the sample current voltammograms had sufficient sensitivity to detect the impact of the anions on the oxygen reduction reaction. And when she analyzed first, what you see on the sample current voltammogram, that the wave recorded in presence of perchlorate occurs at more positive potentials than the one obtained in sulfuric acid. And that's already evidence that the strong concentration of sulfate is inhibiting the oxygen reduction reaction. And then there's also some evidence in the Tafel plots taken from the analysis of the sample current voltammogram. So these experiments demonstrate that the sample current voltammetry has sufficient sensitivity to reveal the inhibition of the oxygen reduction reaction by the concentration of the sulfate anions. And Sibel took the experiment further by comparing the sample current voltammograms between the platinum foil, the polycrystalline platinum foil, and a single crystal of platinum, platinum 110, which was grown by pulse laser deposition on an MgO 110 single crystal. So again, she had to use this cell. The MgO is insulating, so this experiment is only possible by accessing the surface of the electrode from above. And this is clearly an example of a material that can't really be turned into a rotating disk electrode. So this is to illustrate the use of sample current voltammetry as an alternative to the rotating disk electrode. And the experiments up here were sampled at 39 milliseconds, which corresponds to, if you wanted to do these experiments with a rotating disk electrode, that would correspond to a rotation of 1600 RPM. And in black, we have the sample current voltammogram for oxygen reduction on the polycrystalline platinum foil. And in red, we've got the sample current voltammogram for oxygen reduction on the platinum 110. There is about 10 to 20 nanometers of platinum on the MgO single crystal substrate. And there is clear evidence here that the oxygen reduction reaction is running faster on the single crystal surface, and the platinum 110 is known to be the most OR active platinum surface on the in sulfuric acid. So, and there is the corresponding Tafel plot here. So this goes to show that with sample current voltammetry, you can perform experiment analogous to rotating disk electrode, but with electrodes that cannot be mounted on a rotating disk electrode or cannot be made into a rotating disk electrode. So this was very encouraging because it demonstrated that the technique had sufficient sensitivity to pick up differences in catalytic activity for these samples. So the conclusion for this part is that the SCV proves to be a good alternative to steady-state voltammetry when the electrode is not suitable, the material is not suitable for rotating disk or microelectrodes. The current voltage curves are sigmoidal for simple, and you can obtain that with simple electrochemical workstations and very simple cell designs. The sampling times can be very easily selected to achieve the same mass transfer coefficient you would get from rotating disc electrodes. And the agreement with the RDE is excellent when you're matching the sampling time to the rotation rate. It is possible to treat the current time data set to extract Kutakiewicz plots analogous to those you would obtain with the rotating disc electrode. But of course, this is a transient technique, so you have to work within the limitation of transient techniques. And in particular here, you must make sure that your shorter sampling time is not too affected by double layer charging processes, oxide formation, oxide reduction, hydrogen adsorption, desorption, etc. But the technique is sufficiently sensitive to reveal differences in catalytic activity, and we took the oxygen reduction reaction as a good case to illustrate the power of the sample current voltammetry technique. I think in the next few minutes, I will just give you a brief overview of what we've been doing recently with Oliver, this time trying to apply sample current voltammetry to reactions involving adsorbed species, but no mass transport. So we are now talking of having an adsorbed monolayer or fraction of a monolayer on an electrode and using sample current voltammetry to investigate the electron transfer process between the adsorbed species. It's a fairly straightforward mathematical model. We assume that both both reactant and product are adsorbed on the electrode surface, and we assume that the electron transfer kinetics is controlled by Butler-Volmer kinetics. And we have gone through the mathematics and solved the equations, and the objective is to see if we can use sample current voltammetry to derive the unknowns highlighted in red, the 4 unknowns, which are the standard potential, the initial coverage of the species, and the rate constant for electron transfer, and the transfer coefficient. So I won't go through all the detail, but these are easily derived expression, and we then computed from these the sample current voltammograms, and this is how they look. So in contrast to the sample current voltammograms which I showed on the mass transport control, now we're getting sample current voltammograms which have a Gaussian shape. So this is current as a function of time for different target potentials, and these are currents or current density as a function of the target potential. So these are the sample current voltammograms, and they have this Gaussian shape, and the magnitude of the current increases as we shorten the sampling time. And so very different shape compared to what we've seen before. And we've looked at how we've looked at current as a function of, so these are sample current voltammograms, and we've done the same for the charge. So these are sample voltacoulomgrams as a function of the target potential. And what we observe is that the sample current voltammograms are a lot more sensitive to electron transfer kinetics than the charge data. And we have analyzed these to try and derive some mathematical, treatment to extract the 4 nerves that I mentioned before. Yeah, I should mention the corresponding sampling times. So we can see that just changing the sampling time with reasonable times, so that's 1 second, that's 300 milliseconds. The sampling time, the sample current voltammograms are quite sensitive to the time in that region. And I wanted to show you the parallel with cyclic voltammetry. In cyclic voltammetry, you control the time scale with the sweep rate. In sample count voltammetry, you control the time scale with the sampling time. So these are corresponding situations. So sampling at 1 millisecond amounts to scanning performing your voltammogram at 25 volts per second or so. And then sampling at 1 second corresponds to 26 millivolts per second. So you can see that these sampling times allow you to achieve the same sort of time scales as you would with conventional cyclic voltammetry. But whereas these sort of time scales are very easily achievable with conventional, almost the cheapest electrochemical workstation, this sort of sweep rate is already difficult to achieve with the cheapest electrochemical workstation. So it seems as if doing the potential step will afford shorter time scale than what would be achievable with a conventional electrochemical workstation in bicyclic voltammetry. Just to finish with this topic, I just wanted to show you the comparison between the voltammograms and the sample count voltammograms. In blue, we've got the simulated voltammogram for the particular conditions chosen for the electron transfer process, and the dotted curve is the sample count voltammogram obtained from theory. And yes, we are able to achieve to extract the 4 parameters from the sample count voltammetry. In fact, when we analyze the data, you just need 2 transients to perform the experiments, and that's enough to derive the information for these four parameters. And the other thing you can do is to perform a global nonlinear regression on all the transients for all the target potentials, and that's also very powerful to determine the parameters. So this is an approach which is an alternative to the classical Laviron technique, and it goes further than what can be achieved with the Laviron approach. It's analysis of potential step as opposed to analysis of potential sweeps, but we can investigate faster electron transfer processes by looking at the current transients than by looking at the voltammograms. Typically, for these studies, you would acquire 1,000 data points over 1 second. That's easily achievable with even very simple electrochemical workstations. But if you want to push, as I showed you before, using a very fast acquisition system and very stable potential stat, like the PG stat 30 with the fast acquisition card, then you can go down to microseconds. To look at the electron transfer processes for adsorbed species, you should really use microelectrodes to perform the chronoamperometry at very, very short times without having double layer distortion. And as I mentioned, we just need 2 target potentials to determine the 4 unknowns. One potential step should be in the potential window identified for the conventional voltammogram, and then the other potential step should ideally be within 120 millivolts of the E0 to determine if we want to determine the initial coverage for the species. Okay, so I think this is pretty much my last slide. So in terms of overall conclusion, I hope that I have shown you that sample count voltammetry, although it's a very old technique, it does offer some interesting aspects. The sample count voltammograms look sigmoidal in diffusion regimes when mass transport is involved. in contrast, when you have adsorbed species, the sample current voltammograms have asymmetric peaks, which look very Gaussian when you have no kinetic limitation, and then increasingly asymmetric when you have kinetic limitations. Normalizing is very interesting, in particular when there is mass transport, because this allows you to reveal kinetic limitations. The interesting approach here is that you can embed a conditioning waveform before each potential step. We've used electrochemical conditioning waveform, but there is no reason why you couldn't use an optical waveform, for example. You could pulse, you could irradiate the electrode surface with a laser, for example, and have a different way of conditioning the electrode if you wanted. And I hope I've shown you that sample current voltammetry is a very sensitive technique, both to electron transfer kinetics, but also to catalytic activity. And it allows you to work on electrode materials that are not suitable for conventional steady-state voltammetry with rotating disk electrode and micro-electrodes. So I just want to thank you all, and in particular to thank Michal for inviting me to deliver this presentation. I hope you've been interested. Thank you.
Thanks, Guy. That was an amazing tutorial. You covered everything I think we need to know about sample current voltammetry. So if you can just stop sharing your slides, and we'll go to the gallery and we'll take some questions.